letter from me today. Overall summary of the year and Christmas greetings. Silence is golden, at least around here. So I'm going to assume everybody saw it, read it, and understood it. The next 2 weeks, we're going to be standing down. We'll be back the 8th of January and continue. Tonight, Mr. David is going to stand up and present DNA and the deeper side of that. And I'm going to follow with work. Nobody's thrilled, huh? Well, we've got a reactor that's in running condition, but it needs some silicon hoses. And you're going to put them on for me tonight. So we're going to go in the lab, and the reactor's up and running. I've been running it this afternoon. It's working great. And so what we're going to go in there, verify it's operational, then bring it down, and we're going to replace the polypropylene hoses with silicon hoses that can withstand a much higher temperature. Fun thing to do. But maintenance is much a part of science as anything else. The goal for the evening is not to give David any blood samples to work with. We'll be working with some sharp things, and the objective is not to cut yourself. Okay, so barring any unforeseen circumstances, hopefully we won't mix any water with the blood, and we will succeed in having the reactor ready to go. I've got a gentleman coming tomorrow at 1 o'clock named Joe Ballantyne. He is a retired Microsoft engineer. He wants to do fusion for the rest of his life. Just wants to be how he retires. So anyway, should be exciting. He's bought a YouTube, not YouTube, eBay. Thank you, Carl. And he wants to be able to test this eBay neutron detector he bought. And we happen to be the only folks around with a spare neutron or two. It is kind of hard to find. I will tell you that, you can go out there and you can find watch dials and you can find all kinds of interesting things that click and sputter on an ionization meter, but you're not going to find a lot to work with when it comes to neutrons. That's kind of a specialty item here. So Joe's coming in. We've got to have the reactor ready for him. I'd like to be able to run it at high heat. Can't do that unless we have the silicon hoses on. So we're going to put the silicon hoses on tonight. Okay, so David, you're up, and I embedded your PowerPoint video. Go one slide beyond the link, and you should be able to find the video and click on it.
Thank you. Good evening, everyone. Before I get started on this, I wanted to mention that when we were doing liquid chromatography, I was asked a question, and I love it when students ask questions. Please always feel free to do that. That's why I teach. And there's a question about, since we were working with water, it was based on polarity of the substance, and the question was asked, is anything more polar than water? And the answer is yes. There's both hydrofluoric acid and ammonia. They're more polar than water. However, hydrofluoric acid is actually a gas. It's not, when it's not an acid, it's a gas. You dissolve it in water, it becomes acid. If it comes out of the water, you've got a serious problem. The gas is not going to stay on the piece of paper from paper chromatography. Plus, hydrofluoric acid is one of the nastiest acids, and it will eat through the paper, so you wouldn't be able to do that. So, for the purposes of liquid chromatography, water is the most polar one. Okay, just wanted to address that. Anytime you ask a question, if I don't know the answer, I certainly will try to find out in order to let you know. So tonight we're going to talk about DNA, deoxyribonucleic acid. Can everybody hear? Can anyone not hear me? Okay. A little bit audio problems first. So we're going to begin by looking at the structure of DNA, and then we'll look at the replication of it. And DNA has often been called the double helix of life because it's in the shape of a double helix. DNA is what carries the inheritance from the parent organisms. And it's one of the characteristics of life that we had discussed the last time. It's found in both prokaryotes and eukaryotes, and us. Its structure was determined by James Watson and Francis Crick in 1953. A book was written by Francis Crick. James Watson was an American, Francis Crick was British, and Francis Crick wrote the book, The Double Helix. And it tells about what happened from his point of view. There was actually a lot of conflict between the two men. So he gives his side of the story. Now their work was based on work done by a lady named Rosalind Franklin. And she had previously used X-ray diffraction techniques to determine that the basic structure of DNA was made of repeating segments in a helix. That was as far as she was able to go. Another professor worked with her. His name was Maurice Wilkins. Later, Watson and Crick and Wilkins were given the Nobel Prize for their work in determining the structure of DNA. And Rosalind Franklin didn't get it because she made the mistake of dying. The Nobel Prize is never given posthumously. The idea of the Nobel Prize is that you receive a great deal of money and then you use that to further your research. Well, if you're dead, you can't further your research. And so they don't consider people who have died. It's a real shame. So there is a model of DNA, greatly enlarged. And picture of Rosalind Franklin and Maurice Wilkins. This is the original image that they got using the X-ray diffraction pattern with the DNA. And they were experts at that. I don't know how they can tell from that that's a helix, but they did. So, the basic unit here is called a nucleosome within the DNA. It's the simplest package string of DNA. There's 146 base pairs of DNA wrapped around one histone octamer. Let me just see if I can get rid of the reflection on there. An octamer, octa means 8. So there's 2 copies of 4 core histones in it. Histones are proteins. And then the DNA length varies with different species, and it tells you nothing about the species. There are very, very simple organisms that have very, very long DNA, and very complex ones that have very short. The core, the DNA is associated with that histone octamer, and the linker for that DNA, the histone octamer, can be 8 to 114 base pairs. So the histones are the protein molecules. They cause the supercooling of the long DNA, supercoiling, because realize that long, we got miles of DNA inside of you, and that has to get all coiled up really, really tiny and fit inside the chromosome. I'm sorry, it is the chromosome. It fit inside the nucleus of the cell. So that's what's called supercoiling. And the histones are what allows that to happen. Chromosomes are extremely compact. There are a lot of different models drawn to show DNA, and I'm going to show you a number of them tonight. So there's the double helix wrapped around. And then when that supercoils, you end up with that chromosome. It has 2 sets of arms on it, and it's joined in the middle. And when you're trying to separate chromosomes, for instance, in a karyotype, you look at all the chromosomes from one cell of a person, and you take pictures of it, and then you separate them out. And you identify the chromosome by where that place is that they're joined, what's called the centromere. The fact that the arms are off and different, that doesn't matter. You can't use that. The length of the arms from the centromere, and that is basically which helps you to figure out the chromosomes, identify them. So here we have a diagram. You see the DNA, and then it goes in closer, and you see, right there, the nucleosome, and then you have what's often called a string of beads, and that gets coiled up and coiled up more. If you uncoil it, then you start getting the different parts. The bottom picture is the same as the top, it's just pretty. Okay. So Martha Chase and Alfred Hershey did an experiment, and it's famously called the Hershey and Chase experiment. They did it back in 1953. Alfred Hershey, was a professor. His technical assistant was Martha Chase. And I think he must have been a really great man, because he gave her credit. In 10 years of research, I never got my name on a paper. And that was very disappointing for me. My wife did 1 year as a research assistant, and she got 2 papers out of it. It just varies with who you work for and what happens. Anyway, what they did was show that DNA was the part of the whole DNA, which is proteins, the histones, and the nucleic acids. That it's the nucleic acids of the DNA that carry the genetic information for the cell. That tells the cell how to do everything it does. So in their experiment, they used the T2 bacteriophage virus. This is my favorite virus. It kind of looks like a rocket ship. You can see the what they call the capsid up here. That contains the DNA, and then it has what look like legs. It lands on the cell, those attach to the cell, and then the DNA gets injected into the cell. For their research, they were using the T2 bacteriophage virus, and they used that to infected Escherichia coli bacteria, or E. coli. They radio-labeled the different parts. DNA was labeled with phosphorus 32, a radioactive label, and the protein was labeled with sulfur 35. And then they went ahead and injected, or had the organism, the virus, inject into the E. coli. Then they centrifuged it. The protein will pellet at the bottom because it's much greater density. And in the supernatant, the liquid in the top of the tube, you have the DNA. And what they found was the fraction with the DNA was what ended up in the virus, making the difference. And so they were the first ones to determine. Before that, there was a lot of arguing, and most scientists, including Hershey, thought it would be the protein because proteins are so complicated, so complex. But then we found out the DNA is actually pretty complicated too. So deoxyribonucleic acid, or DNA, it's a long molecule, spirals in the form of a double helix like a twisted ladder. Took a ladder and just twisted it around, then that's what it would look like. The sugar in it, deoxyribose, if the molecule ends in OSE, it's often a sugar. It's a 5-carbon sugar. It's in a ring. And then you have ribose sugar, which is exactly the same as deoxyribose, except that it's lost oxygen. Deoxyribose has lost an oxygen. So that's the only difference in the structure of the two. As you see in red, deoxyribose, the hydroxyl OH on ribose has been changed to the hydrogen on the deoxyribose. And on the side there, you can see the twisted ladder shape, and the color coding for the rungs of that ladder are the nucleic acids. There's 3 different nucleic acids. There's adenine, thymine, guanine, and cytosine. Those are the only nucleic acids found in DNA. Only 4 of them. And yet you can get all the different combinations that go for everything from a little bacterium to you. The sugar molecules in the model alternate with the phosphate groups. The phosphate groups are what you see in the, I guess that's like a purple there, lavender. I'm not very good with colors. But that's the phosphate group. That's the sugar molecule. In this case, it's ribose, so that's an RNA. And then you have the base, one of the 4 nucleic acid, although with ribose, it's different base. So DNA is made up of 4 different nitrogenous bases, as we said. One group are called the pyrimidines, and the other are called the purines. You always have a pyrimidine attached to a purine. If you didn't, it would throw off the structure of the ladder because they're different sizes. And as it shows over in the left corner there, the pyrimidines are abbreviated C and T for the cytosine and thymine, and the pyrimines are abbreviated G and A for guanine and adenine. So when they're bonded together, it's hydrogen bonds that are holding them together. Much weaker than covalent bonds, which we're going to see when it comes to replication. That's what makes it possible to replicate them, because it's easier to break those bonds between the nucleic acids. There's 2 of them, 2 hydrogen bonds between A and T, and 3 between G and C. And they're always in those combinations. It's always A to T and G to C. So again, there's the nitrogenous bases. The ones here in the colors are the nitrogenous bases. The blue is the sugar, 5-carbon sugar, and then the phosphate group is shown as the phosphorus and oxygen. And that gives you your base pairs. The distance it goes before it twists is always the same. It's a certain number of base pairs. And that's called the major groove, and then the other one's the minor groove. And then the whole backbone, not the rungs, but the outside part of the ladder, is the phosphate combined with sugar groups. Oh, I wanted to mention one other thing on that slide. Carbon, anytime you have a ring molecule, or well, any carbon molecule, the carbons are numbered in a certain way. And I don't want to go into depth on that now, but the way it works out, when you have adenine, that is going to be a 3' end on the strand of DNA. And you'll notice that the complementary strand, on the left, we have A at the top, adenine, and then we have thymine to the right of it, and that thymine makes that a 5' end. Because they're complementary, you always have this, what's called an antisense to the DNA. So the 3' end and the 5' end, you're going to see later that's very important in the way replication occurs, and it actually causes some problems. So, DNA replication. We've gone over the structure. Now we're going to take a look at the replication. And there you can see the whole thing. Memorize that and you've got most of it right there. But let's go over it. Wrong way, David. There's something called the central dogma of biology. And that central dogma is that information flows from genes to proteins. And so the genes are going to replicate, and then there's a process called transcription, where the DNA is a template for forming a particular RNA, and then by translation, that is turned in by ribosomes turned into proteins. And so all the information contained in the DNA goes through those processes so that you end up with protein. Why does the DNA need to replicate? If you're going to reproduce, then you've got to have more DNA than was originally in the cell. And so that DNA can be passed on. And so it makes copies. I divided this up into 3 stages. Some people put it as 2 stages. Some people put it as 5 stages. It doesn't really matter a whole lot. And I don't like people trying to memorize things like which stage is which, because when you get to things like mitosis and meiosis, it's all a continuous process. It doesn't go a little way, stop, and then you have another stage. Everything's happening at once. But the 3 stages are the separation or the unwinding of the DNA strands, the synthesis of what's called an RNA primer, and we're going to take a close look at that later, and then the copying of the template strands. All of these things have to occur for it to work properly. So in the first stage, separation, the DNA is unzipped. The first stage of DNA replication, there's an enzyme called topoisomerase. One of the things you'll notice is that in chemistry, OSE at the end of a word means sugar. ASE at the end of a word name means enzyme. So topoisomerase is an enzyme. And it's the enzyme that unwinds that double helix. Because it's got to be unwound before you can start doing anything with it. Then you have another enzyme, DNA helicase, ends in ASE. That breaks the hydrogen bonds between the nitrogenous bases. And where that happens, you end up with what's called a replication fork. So you have the regular DNA over here, and then it gets split up by the helicase after it's been unwound by the topoisomerase, so that you have single strands of DNA out here in order to replicate it. I really like this diagram because when a lot of teachers present this, my professors when I was in college, they would get you thinking that the DNA separates and you got this long strand. No, it's actually happening in bubbles, in a whole bunch of bubbles. There's a lot of bubbles where they have the replication going on, and it's within those bubbles. And so every one of those where you have this separation, that's called a replication fork, where the replication is occurring. And you end up forming new DNA. So that was the first stage, unwinding, separating. In stage 2, it's synthesis. Synthesize something means to make something. The now exposed nitrogenous bases, AT and GC, can now form new base pairs using bases that are just floating around in the nuclear matrix. All of this is occurring inside the nucleus. And every time you have an A, it's going to attach a T to it. And every time you have a T, it'll attach an A. Every time you have a G, it's going to attach a C. Every time you have a C, it's going to attach a G. So that's going to happen. However, you have to prime the original single strand of DNA to get the replication to start working. Well, how do you prime it? Oh, wrong way again, David. Okay, I'm going. There we go. We have another enzyme come in. Now, this is called RNA PRIMASE, and it adds short primers that are only about 11 base pairs long to part of the single strand of DNA. It's making, because it's an RNA PRIMASE, it is making a short segment of RNA. Now, the difference between RNA and DNA is just one thing as regarding this, and that's that you no longer have thymine, you have uracil. So instead of A to T, it becomes A to U when it makes puts on the primer. So that's done by another enzyme, RNA PRIMASE. And once that's done, then the strand is primed in order to add more bases. And now we're on stage 3. In stage 3, you have the actual copying, making of the new strand of DNA. And this is done by DNA polymerase, number 3. There are different DNA polymerases, which is what kind of a molecule?
I'm not sure if this is
Yeah, if it ends in ASE,
enzyme.
it's an enzyme, okay? So DNA polymerase 3 is another enzyme. See how important enzymes are for everything you do. it continues adding more bases. But instead of adding U, like RNA does, it now puts in T. So it's adding what we need in there. The new DNA, the new strand, single strand, is complementary to the original. And so that's formed. And that's what's going on in this diagram at the bottom. But there's a catch to it. Very, very important one. DNA polymerase 3 only works by starting at the 5' end. It can't start at 3' end. That means that you've got this strand that's separated, and you've got the replication fork. The one strand is going to have the 3' end away from the replication fork. And the polymerase will then start at the 5' end and just start copying. So you only have to have one RNA primer there, and it'll keep going. But the other single strand of DNA is backwards to that. So the 5' is at the other end, away from the replication fork. So what happens is that DNA polymerase will move away from the replication fork, find the primer, and start producing the copy back towards the replication fork. And then when it gets to the end of that, it jumps to the next RNA primer. So the RNA primers are strung all along that one single strand. That single strand is called the lagging strand. Because of this process, and there's proteins that come in and hold it in the right place and make sure that the DNA polymerase goes where it needs to go. But as we're going to see, that slows things down. The lagging strand is what we call discontinuous because it has chunks where it's made DNA. And then it has other areas where it has a short bit of RNA, like 11 base pairs. The chunks that are made are 150 to 200 base pairs. And they're called Okazaki fragments. And you might guess the man who discovered this was named Okazaki. It was he and his wife, which is kind of fortunate because you don't have to know two names. All you got to know is Okazaki. That was the name for both of them. So we end up having these Okazaki fragments. They're not joined together properly on that lagging strand. As that lagging elongation occurs, another enzyme of DNA polymerase 1, before we had DNA polymerase 3, now it's DNA polymerase 1, and it replaces all the RNA primers that are on that lagging strand and the one that's on the leading strand. And it takes them, all the uracils and replaces them with thymine. Where they get the thymine, it's just in the nuclear matrix, floating around. And then DNA ligase, what kind of a molecule is that?
Another enzyme?
Ends in ASE. That's another enzyme. The enzymes are what control this mechanism. That DNA ligase, to ligase things means to join them together. So it joins the different fragments together so that you end up with a continuous strand of DNA on the lagging strand. No. Then another enzyme, RNA PRIMASE, adds short primers. Something went wrong. I'm going the wrong way again. There we go. Okay. The replication of the lagging strand. As I said before, it has to go through all these different movements with different enzymes. That makes it a slower process. It's the rate limiting factor in replication, slows it way down. And because of that, mistakes happen somewhat frequently. You might get mismatched pairs. You've got an adenine attached to a guanine. Can't have that. So DNA polymerase 3, again, one of the enzymes we already talked about, does a process of proofreading. As it's replicating, it's looking at the different pairs and correcting them. So if there's a uracil in there or a G attached to it, an A, then it's going to remove one of them and replace it with what it should be. A very important process, because otherwise you've got a mutation. And most mutations, by far most mutations, are harmful, if not downright fatal to an organism. And that's what we see here in the diagrams. It also will correct other problems that it finds, like if there's a place where they're not joined, get that done. And some people have shown that DNA polymerase 1 can also do that. There are differences in the replication process, not very major ones, but some, between prokaryotes and eukaryotes. And I just didn't want to go into all the details of that. Books get written on that sort of thing. So the end result of this whole thing, with all these enzymes, is making an identical strand of DNA. Each one that's produced now is an antisense, meaning it's the complementary one. And since you've got two strands that are being produced, and they're antisense of the other ones, when they're done being made, you end up with identical to what you started with. And we've got a video that summarizes this, if it works. How do I activate it, Carl?
Click one more slide.
It's this slide at
Click.
the very bottom.
Click next. Move forward.
Aha.
Now click on that.
If I
Nope.
click, it goes to the next slide.
Hang on, let's see if I can
I
reach it.
think you have to click on it.
Okay, hang on.
The Amoeba Sisters have done a variety of biology ones. And
Waiting.
DNA. We talk about it so much. It's the ultimate director for cells,
Can
and
y'all
it
hear
codes
that?
for your traits. With a molecule that has a function like that, it makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where. Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when. When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented. I'm going to introduce the key players in DNA replication so that you can get some background information. Now remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helicase, the unzipping enzyme. If you recall that DNA has two strands, you could think of helicase unzipping the two strands of DNA. Helicase doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. PRIMASE, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. PRIMASE makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and exceptions to every biological process that we can't include in such a short video. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that enzyme's name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next steps. PRIMASE comes in and makes RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of this ring structure. Now, you do the same thing for the other side, but keep in mind DNA strands are anti-parallel to each other. So let's count these, again clockwise after the oxygen. 1', 2', 3', 4', 5'. And the 5' is out of this ring. This strand on the left runs 5' to 3', and the strand here on the right runs 3' to 5'. We'll explain why all that matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. PRIMASE places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is, when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase once again is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one. One last thing. Surely you've had to proofread your work before to catch errors. In this process, you don't want DNA polymerase to make errors. If it matches the wrong DNA bases, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein, or no protein. DNA polymerase is awesome. It has proofreading ability, meaning it rarely makes a mistake, which is a good thing. So remember how we said there is far more detail to this process to explore? The detailed understanding of DNA replication has led to some life-saving medical treatments that can stop DNA replication in harmful cells, including pathogenic bacteria or human cancer cells. We encourage you to explore beyond the basics. Check out the further reading suggestions and the video details to explore more. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
It's not advancing for me.
Okay.
So that's a good, simplistic way of seeing it. Helps when it's an active thing. Once we get this forward, there's just one more slide.
Hang on.
There we go. And these are just two others related. I'm not going to show them to you. One is the crash course, where Hal does a great job of explaining things, and there's crash courses on everything. The other is actually the story of Rosalind Franklin and how she missed out on the acknowledgments that should have gone to her for the work that she did. It's a true shame. So why did we bother to learn this or go through this? That's because last time I showed you we got in gel electrophoresis equipment. Today, we got all the materials needed to use that, to separate DNA. So in January, we're going to do that, and we're going to solve a murder mystery using DNA fragments, okay? So that's something for you to look forward to. Any questions over any of this from anybody? You probably learned it all in, what, junior high school? Maybe? All right, thank you.
Okay. Can everyone hear me? Okay. Um, so we're going to attempt to journey to the nuclear reactor. We'll get set up there to do some work um, in the hoses. We're also going to make sure that we have the number 10. So if you'll unplug from your air masters and come on into the reactor room, we'll get started. Okay, could I get you to go get a couple of more stools out of the lab and roll them in here?
That in for me. Yeah.
Okay, that doesn't work for you. I'm going to need you.
Okay. Turn that up.
It's fine. Okay, all right. Okay, so I have a seat here. Talk about what we're going to do. Now, can you folks at home see us?
Yes, we can.
Good. Okay. Um, so what we're going to do, okay, we just jumped the power on. Okay, we're running about 70,000 volts right now. And you see the meter. As you can see, it's pegged out right away. I have to go down a notch. Okay. 19,000, and right now, if you take a look at the counter, we're up over 2.5 million neutrons per second. So this is a extremely high level of output. And I'm seeing 1,500, 1,900. This is through the shield. 1,900 counts through the shield. So very high level of radiation. We're going to drop the radiation down here substantially. Now I've dropped the high voltage. I'm going to turn the gas off.
I'm not sure if this is the appropriate time to ask this, but I was wondering if you would quickly go over how we can calculate the REM based on the neutrons you can sense that are coming out of the shield.
Okay, 333 counts equals basically 1 rem. So you're looking at 5 millirem, okay? Since the meter counts, 333 counts equals 1 millirem, we're looking at about 5 millirem on the surface of the shield. But the thing that's important to note, and we'll take a look at this again, power, main page, go back to gas, bring our gas up, main page, high voltage on. Here comes our radiation again. Now again, we're looking at about 68,000 volts. We'll take a look at our meter again. We're 400, 700, 800, 1,300, 1,100, 1,300. So say about 1,300 right now. Give it a little bit more. Okay, there's 1,500. So at 300 a pop, 3 5s are 15, say 5 millirem, right there. But here's the deal. If I move literally 1 foot away, that drops to 24 total counts. So you're looking at the fall off is virtually out here at the safety line. We're down to below 100 counts, or less than 1/3 of 1 millirem. And 1 millirem is 10 microsieverts, so you're looking at about 7 microsieverts of neutron radiation. 7 microsieverts, you're allowed 500 millirem a year. And when we talk about millirem, I say, okay, that's 5 millirem. 5 millirem of what? 5 millirem an hour. So what's happening here is when I say I've got 5 millirem on the shield, that means I have to stand here for an hour to have an absorbed dose of 5 millirem. As an adult, I'm allowed 5,000 millirem, or 5 rem per year. So I would have to stand here for 1 hour to get 5/1000ths of my annual dose. So we're really not dealing with a significant amount of radiation from a health and safety aspect. But we are dealing with an amount of radiation that is completely adequate for the kind of research that you can do in simple biology and chemistry, oncology, a whole raft of material sciences and imaging, just a whole bunch of fun things.
I'm just a bit confused about the per hour. It doesn't the Geiger counter count in per seconds? So if there are 333 counts is to 1 millirem, and there's 1500 counts per second.
It's giving you the millirem, but it's giving you that millirem not per second. All radiation is measured over an hour. What they don't want to do is give you a meter that you have to stand there and hold for an hour in order to determine the radiation. So what happens is they give you a meter that's prorated. It'll give you the amount per hour that machine is putting out, but it'll give it to you in 10 seconds or 5 seconds or 1 second, depending upon what you've turned it on. Right. So when I hold this machine up, you can see that it fairly quickly, about 1 or 2 second intervals, it's updating. But it's updating the number of millirem you receive per hour. So, yeah, if we were anywhere near a millirem per second, we wouldn't be walking away from this. So, and we'd also have discovered fusion, and everybody would also be very wealthy posthumously. So anyway, we're going to close her down now. We got the gas off. We're just going to run some cooling pumps so everything's kind of on the ready, but there's no way that this system can actually light off when the master control is down. So now we'll raise the lid. For those of you who have not heard this spiel, this lid weighs over 1,000 pounds. It has 55 gallons of borated paraffin, 66 cadmium plates, and a lead inner wrap to prevent the X-rays from getting out. The machine puts out a good rem of X-ray when it's in operation. And the X-ray is actually far more dangerous than the neutron radiation. We have to get very, very careful when we're working with that. Okay. Now I just shut the refrigeration unit off. And that does two things. It not only stops the refrigeration, but it also stops the pump. And we don't want the pump pumping coolant up around the pipes when we're busy changing the hoses. That would be a bad thing. This is the plumbing we're going to use tonight. And how are we doing here? Okay. So I'm going to need some tools from the toolkit. You know where the tools are. So I need a pair of wire cutters. And I need the blue-handled razor knife that's up in the top right-hand drawer. Okay, so Evan's going to get those things for us. And this silicone is much more supple than the PVC. PVC becomes quite rigid. Polyvinyl chloride does not like cold. It gets very stiff, hard to work with. Now, in addition to everything else, this reactor has an interlock that prevents the high voltage unit from powering on in the event the lid is raised. So, at the present state, the high voltage interlock is disengaged and the power supply cannot come on. We've got it shut off at the panel, and we've also got it shut off at the power supply. So three levels. Some of the features of this, these are called the conflats. This white thing up here is the high voltage electrode. And this is the X-ray detector. And this guy back here with the red stripes, he's our neutron detector. Hey, Evan, you want to click on your microphone for a second?
Yeah.
So you having trouble finding it?
The wire cutters, yeah.
Okay. Extreme left-hand side, third drawer up from the bottom. Should be blue handles.
This one?
Perfect.
And then right east.
Okay. Alrighty. So I'm going to do one of these, and you'll all kind of get an idea for what's going on here. I'm going to be sending somebody else out to get some tools. Caleb, you know where some stuff is, right? Oh, okay. Well, this is very challenging. I need a roll of paper towels. And the towels are on the outside of the toolbox, and they're held on with a magnetic holder. So just pull on the holder, it'll come right off, grab the roll and bring it in. And that's in case we spill some water. What? The ones we're going to be working with initially No, but at some point, yeah, we will. Okay? Perfect. Thank you. Okay? This is in case I get sloppy. Okay, this is our hose. Now, I'm going to begin out here on this side. I just want to kind of forewarn everybody, there's a possibility there are 3 copper heat sinks on the side of this conflat. They theoretically have been applied with epoxy. They've been baked on up to 160 degrees. They should be solid as a brick. However, I have seen these things come off with very little force, all by themselves. If that happens, it's not your fault. Don't blame yourself. These things aren't always stuck on as tight as they look. Now I've got a piece of tubing here. Can you folks on camera see my finger where I'm pointing?
Relatively, yeah.
Okay, so there's a hose here, and it comes out of one heat sink and it goes into another heat sink. But any heat sink that this PVC is in contact with can't go above about 130, 140 degrees, or the PVC will melt. The silicone is good to much higher temperatures. So, what do you think is going to happen, since these are all full of water, the moment I cut into one?
It's going to leak.
Actually, no. And that's counterintuitive, and I'll tell you why. The pump is down here on the floor, number one. Number two, we've turned it off. So both of the water lines that go down to the main pump and to the tank underneath no longer have any pressure on them. As a matter of fact, the gravity is pulling the water down, and because there's no air in the line up here, the suction, okay, is holding the water in stasis. The moment I open an air gap up here, what's going to happen is all that water is going to run back down inside to the tank, and if the tank didn't have enough room in it, it would flood. But it does. So, we're going to pop the lid on this thing. We're going to pop the hose on it. Instead of water spurting all over every place, you're going to see air flow into this, and you're going to see all the liquid come out of the lines. And what's happening is the water is being dragged back down to the pump. Okay, so here we go. What I'm doing...
Before you start the process, could you zoom into that region?
I can't, because I'm here and that switcher's in there, and I'd have to unhook and go all the way in there, and yeah.
Okay, that's fine.
Unfortunately, tonight is not a good night. Hopefully, over the time Christmas break, I can take care of some of these problems, and I will hopefully be able to do exactly that. I just didn't have enough time tonight. So here we go. We're going to open this guy up, and what I'm doing is I'm cutting into the PVC, and I see air bubbles in it right now. And it's just far enough to to weaken it so that it'll open up. And all of a sudden you can see there's not a drop coming out of this thing, and the water's all flowing back down the line in the other direction. So now I'll cut the bottom one off. And the thing that's nice about rubber gloves is, think of it this way. I'm already wearing Band-Aids. It's just a joke. Anyway. So, as you can see, the bubbles are still moving, sucking all the liquid out of the way. Okay, now I've got a piece, and this was a piece that went from point A to point B, and I'm going to replace it with a piece of my silicon. So, I got my chunk of stuff here, and I'm going to measure off approximately the same length. I'm going to take my wire cutters and cut that piece of silicon off. Now, I'm going to pass these around for a moment so that everybody can kind of see the difference in the texture. Go ahead and give one a squeeze and the other one a squeeze. And you can see they're really different. PVC is very hard, very inflexible. The silicon is very flexible.
Why is the PVC really discolored?
There was some bacterial growth in here, and we threw in some chlorine, and so the chlorine I threw in was just a little too much, and it interacted with some metal down in the junction here and created some rust, and the rust has colored the water. So we have a combination of things going on. We have some rust-colored bacteria. Those are additional maintenance items. We'll get to them. Okay, so now I'm going to take this piece of silicon. I noticed here that this is not perfectly straight. Well, that was the way it came from the manufacturer. So I'm going to take a moment and just make sure, cut this off with a dikes one more time to get a nice straight end on that. We'll go ahead and we'll push this guy on. There he is. He's on there. And now before we go any further, I'm going to go ahead and turn him back on. And we'll make sure that he doesn't leak, because I need to know that now. So you see everything flying around. A few air bubbles. But it looks like it's a fairly tight connection. I think we have a winner there. Okay. So now we'll go ahead and shut her back down. And that's the routine I want to go through tonight. We've got plenty of connections on this side and on that side to do. So we're not going to get them all done, but I'd like everybody to do at least one without cutting yourself. That's the challenge. We have paper towels here if you do. And my wife's really good with a needle and thread. She can sew your arm back on really quick. Some rules about handling a knife like this. This clicks out and it locks and it's nice and steady. But you don't want to get your fingers up close to the blade, okay? And there's a finger grip right here. So when you're using this thing, you don't get your hand down here and do this. Because if that knife slips, you're going to go right across your other hand. So that's not a good thing. So what you're going to do is you're going to, with one hand and your finger on the grip, you're going to cut through the PVC and you're going to cut lengthwise. Now, where's the piece we had? I want you to look at it. What I did was I just slit the end open. You see how that is? Just slit that end open a little bit. And once the tension was off of there, it was very easy to bend it and rock it off. So you just want to cut a slit in each side, like that. Okay? So that's where we're trying to go with this. All right. Evan, you're up first. Ever done anything like this? Oh boy. Okay. So the hose I want you to go after is going to be this guy. And I note that he's got a clamp on this side. So obviously he's had a leak problem before. I'm not surprised. I suspect we probably overheated the reactor and didn't. All right. So I want you to do this side. What you're going to do is you're going to go right there and you're going to cut right there. And I don't want you to use a lot of pressure because that might just rip the heat sink off. But I want you to make several cuts in the same place, each time a little deeper until you think you're down through the material. Okay? And keep your hand out of the way while you're cutting so that if the knife goes like this, you're not going to stab yourself. Just apply more and more pressure all the way back to the solder joint. You're starting up to, you want to go right up against the metal on the thing. You're starting out way too far. You want to go all the way back here. Okay. It doesn't matter if you have to make several cuts. Just work it until you get all the way through that first layer of polyvinyl chloride. When you start seeing air bubbles, you'll know you're through it. Okay, there comes the first air bubbles.
So I just like.
Okay, so now slowly work it with your fingers. Try not to put too much pressure on it, because again, we don't want to rip the thing off. And see where it's not yet cut. Okay? You're getting the hang of this, I can tell. Okay? got to be close there. Okay, just pull on a little bit. There we go. All right, now let me get you a screwdriver. We're going to do the other one. I'll be right back. Okay, so here's the screwdriver. You're going to take that clamp off first. So you got to see where that screw is, and then get yourself around on the side of the reactor where that screw is, and go ahead and take that clamp off. Can I make you in charge of the tubing? Okay. Now you're going to unwind that guy until you can slide him down and off the other side. Okay. And work him through. Okay. Now go ahead and get your knife and go to work on the second side. Give it some thought. I don't want your two hands that close. I think that's probably the best right there. Just take your time and work it. This isn't a race. Okay, let's flex a little bit and see how much of it's split. Okay, that way. Oh, there we go. Yeah, you had it already cut all the way through. Okay, so we got that one out of the way. So here's your wire cutters, and you're going to need that for your length. Make sure you get it just the same length. You're going to have to kind of bend it around and get really close. If you're within a 1/4 inch, it's fine. Yeah. Woohoo! Look at that. Nice and straight. It looks fine. Looks absolutely fine. Go for it. Okay. Okay, looks tight. We'll go around here and throw the switch. See how you did. Want to dance? Okay. You see those air bubbles? We just give them a tap, and it usually causes them to go away. Okay, so again, looks like we have 2 of these replaced already. And things are going well. Good. All right, do we have enough to do one more? Looks like we got one more, and then we're done for the night. Who's brave enough to jump up here? Come on. Okay. Now, okay, so this is the one I want you to go after. You're going to open this one up first, and then when you're done, you're going to use the screw clamp, and the screw is right through there, so you're going to have to go in there and unscrew him, and then slide the clamp back, and then you're going to have to get in there, and what'll happen is you can just, without cutting him, you can just pull him right off the end of that copper tube, because what's holding him on there is the hose, or that hose clamp. So we're just going to cut one end this time. Right there. Right. Perfect. Just take your time. Apply a little more pressure each time as you get better control of the knife. Your confidence will increase. There's the first air bubble. Looky there. Okay, keep cutting. Okay, let's put the knife down and then wiggle that back and forth and see how big of a split you put in it. Got a pretty good size split in it. Now what you can do is you take that screwdriver and put that blade into the slit and then kind of twist it and try to open it up a little bit. Yeah. Now pull back on that guy. There we go. Okay. Super. Now go ahead and unscrew that inner piece. We may get a few drips on this one because there's water in the hose. Okay, and try to slide that down the hose. Probably need to untighten it a little more. Okay, that should do it. Let's see if we can slide it off now. Okay, now wiggle the hose. Come around here on this side and grab the whole hose and try to wiggle that guy off. Take the hose and twist it like this. Twist it and pull it at the same time. And work it down. You see it coming? There we go. Woohoo! Okay, so we got a little bit of paper towel work here. Okay, we need the hose. Now you're going to go for something that length. Okay. think that's a real good guess. RWB, okay. All right. You can start there. Yeah. Let's see if that feels. Does that feel loose or tight?
It seems a little looser than the other one was, but maybe that was just because it's been on there, but for a while.
Feels looser.
It's easier to get on than it was to get the other one off.
Okay.
Oh, okay. Actually, it seems okay.
Okay. Let's go ahead and connect it up.
Okay.
On the other side. Okay. Now what I'm going to do is I'm going to turn it on just like that, and you're going to watch and tell me if we got a leak.
Okay.
Here we go.
Nope, we're good.
Okay. Like that story. This stuff is more supple.
Yeah.
And so it takes a grip a lot easier. Okay. I think we're there. You kind of see the process. Hopefully we can get the rest of this done over Christmas break. If I don't, you guys will be doing it in January. I'm going to go out and stage the folks tonight. Thank you for joining Northwest Nuclear Labs. We'll see you on January the 8th. Thank you. Have a great, happy Christmas and Hanukkah.